Chapter 19 covers both processes of cell division. So mitosis, the process that we covered in the last video, where the goal is to create two identical diploid somatic cells. That's the process that is happening most in our bodies, replacing old damaged cells and repairing tissues, mitosis. But we have this other process of cell division called meiosis, and the goal of meiosis is to create 4 genetically different haploid cells. So this is how we are creating those gametes, sperm cells and egg cells. You're going to see when we go through the phases of meiosis that there's a lot of similarities between the two types of cell division, but it's really, really important that we know that there are those major differences. And the major difference is that at the end of meiosis, we're going to have 4 cells that are genetically different. So we're going to go through a couple of processes that kind of recombine our DNA and mix it up a little bit so that we can have a more genetically diverse population. And that's one of the advantages of sexual reproduction over asexual reproduction. So last time, and when we looked at the reproductive system, we looked at spermatogenesis, that formation of sperm cells, and their progression from spermatocyte, the diploid cell that's been differentiated to later become a sperm cell, to spermatid, a haploid cell containing a single set of DNA. The formation of this haploid spermatid was meiosis actually happening. Then we know that the aim of meiosis is to create haploid gametes that provide only half of the DNA necessary to code for a new individual, because half will be donated by the sperm from dad and the other half by the egg from mom. By doing that, meiosis keeps the total number of chromosomes in a new individual constant from generation to generation. And as we see when we'll discuss the phases of meiosis, those processes also increase genetic variability within the population. That's one of the advantages of using sexual reproduction over asexual reproduction, where you're just creating another copy of yourself and creating a new organism, like you see in many protists or plants or bacteria sometimes. But we obviously do not have the capability of doing that. So we're going to focus in this class on sexual reproduction. In mitosis, we saw that the end result was 2 cells that were identical to one another in every way. They had the same amount of DNA, they had exact replica copies of DNA, and they had the same number of organelles. After meiosis, though, the end result will be 4 cells that are genetically different from one another. So you'll definitely recognize the phases because they're the same as mitosis. But meiosis is going to involve 2 individual rounds of cell division. After meiosis 1, the first round of cell division, we're going to be left with 2 cells that each have 46 chromosomes, so still in our 23 pairs. After meiosis 2, the second round of cell division, those sister chromatids from each pair of chromosomes are going to be separated into 4 cells that now contain only 23 chromosomes, so half of the genetic information. As we go through each of the phases, during meiosis, what you're going to kind of be aware of and mentally kind of thinking about is what is being split in meiosis 1 and what is being separated in meiosis 2. During meiosis 1, just like before you undergo mitosis, cells that are going to undergo meiosis are also in that period of interphase before they enter meiosis. And in interphase, just like we saw before, the DNA is replicated and the copies, those chromatids, remain connected by the centromere. So all of that is the same. And then the first round of cell division, meiosis 1, occurs by the same phases that we saw in mitosis, but with 2 very important additions, and those additions are to produce genetic variability. So you'll see these new events in prophase 1, when crossing over takes place, and in metaphase 1, when independent assortment occurs. During prophase 1, we're going to see the same things that occurred during prophase and mitosis. The DNA condenses into chromosomes, the nuclear membranes break down, and the spindle apparatus begins to form. But since this is meiosis, we're going to see a couple of additional things happening. A process called synapsis occurs, where homologous chromosomes pair up and become aligned with one another. So for each of our chromosomes, we got one from our father and one from our mother, giving us 2 copies of each gene. During interphase, both of those chromosomes were replicated to form 2 chromatids connected by the centromere. So we now have 4 copies of that chromosome. During synapsis, the replicated chromatids from our father and the replicated chromatids from our mother line up together so that each homologous pair of a particular chromosome line up together during metaphase 1. This is a really important step to understand, and honestly one of the most difficult, so make sure that you're reading and rereading this section in your book before you move on. So we're seeing synapsis and crossing over also happen in prophase 1. And crossing over is where sections of DNA from the maternal chromatid from mom and sections of DNA from the paternal chromatid, the one from dad, are switched around among homologous chromosomes. We're going to look at that in a better image at the end of this section. So all of that is happening in prophase 1. During metaphase 1, the homologous pairs line up randomly along that equatorial line at the middle of the cell. And this random assortment, also called independent assortment, is the second way that meiosis results in increased genetic variability. So we had crossing over, which is actually physically recombining the DNA, and then we're going to have independent assortment, where they're going to be lining up randomly along the equatorial plate. So you might get some of the original chromatids from the maternal, or the ones from mom, and some of the original chromatids from dad in each individual cell randomly. Also during metaphase 1, a spindle fiber from one centriole attached to one homolog, and a spindle fiber from the other centriole attaches to the other homolog. So now all of the chromosomes are lined up along that equator, the equatorial line. During anaphase 1, the homologous pairs are pulled toward opposite ends of the cell by the spindle fibers. So during meiosis 1, during anaphase 1, we are separating homologous pairs. We're not seeing any centromeres broken during meiosis 1 because our chromatids are remaining together. The goal of meiosis 1 is to separate homologous pairs. During telophase 1, once the homologs reach their separate ends of the cell, the nuclear membranes begin to reform and cytokinesis begins, just like we saw during mitosis. And once cytokinesis occurs, that first round of meiotic cell division is complete, and we can see meiosis 2 begin. Meiosis 2 looks a lot like mitosis, but results in the separation of the sister chromatids into 4 haploid cells. Each of the following phases occurs in both of the daughter cells that were produced by meiosis 1. So during prophase 1, the DNA condenses again into chromosomes, the nuclear envelope breaks down, and the new spindle fibers form. We've seen that before. During metaphase 2, the chromosomes, still those 2 sister chromatids attached by the centromere, line up along the equatorial line. Seen that before. During anaphase 2, the centromeres break and the chromatids separate and are pulled towards the opposite centrioles. Seen that before. During telophase 2, one complete set of chromosomes, each are reaching the poles of those cells, and then cytokinesis occurs in each cell, producing a total of 4 cells. At the end, each of those 4 cells now contains only a single copy of each chromatid, so they are now haploid cells. Let's look at those processes of crossing over and independent assortment again. Crossing over, remember, happens in prophase 1 after the maternal chromosomes are paired up with the homologous paternal chromosomes. and sections of DNA are physically exchanged between the maternal and paternal chromatids. Only homologous sections are exchanged, and that means that only sections that contain the same regions of DNA that code for the same traits are exchanged. And after crossing over occurs, each chromatid has a new DNA sequence that neither of the chromatids donated from the individual mother or father. That second process that produces genetic variability was independent assortment, which happens in metaphase 1. As the homologous pairs are lining up on the spindle fibers along the equator of the cell, they line up in a random independent order, meaning that it's random whether the maternal or paternal homologs are facing each end of the cell, so which direction they're going to go when they're separated during anaphase. For each pair of homologs, there are 4 possible combinations then in the resulting gametes. Please, please, please spend some time going back and reviewing meiosis 1 with these events in mind and make sure that you could explain how they are resulting in increased genetic variability. This is one of the most difficult topics, I think, to do in an online course like this. So if you're struggling with these phases of cell division, please reach out to me this week, because this is one of the most challenging ones to teach online, because if we were doing this in class, we'd be doing a lot of physical moving around, pretending that we're chromosomes and making sure that we're knowing what's happening with them. But I do have a fun video of mitosis in action. There's a lot of vocabulary when you're talking about what state is the DNA in at each of these phases. So again, if you are struggling with that at any point, I would love to talk to you about them more. I will see you in the next video when we will start talking about genetic inheritance.